Hello, everybody, and thank you for joining my talk on the impact of functional connectivity of the motor system on the resting motor threshold. My name is Melina Engelhardt, and I'm a PhD student at the Charité Berlin. The resting motor threshold is commonly defined as the smallest stimulation intensity to reliably elicit muscle-evoked potentials in a target muscle. On a functional level, it is defined to measure excitability of the corticospinal tract. RMT is one of the most commonly used measures in studies using transcranial magnetic stimulation. It is often used as an outcome to measure corticospinal excitability, for example, to quantify changes due to a disease or effects of an intervention. It is also used to scale stimulation intensity of other TMS or rTMS protocols, and this goes way beyond the motor domain, for example, extending to therapy approaches in depression. This highlights the need for a very thorough understanding of what the resting motor threshold means, what influences it, and what might confound measures of the RMT. One of these factors that has been identified a while ago already is the coil-to-cortex distance. With an increasing distance between the coil and the cortex, also the resting motor threshold increases. Similarly, with an increasing age, the RMT increases. Other factors that have been suggested include sleep or awakeness and white matter properties. There are many other factors that have been researched in terms of contributing to variability in the resting motor threshold, and most recently, functional connectivity of the motor system. In a paper that was published in 2017 in Brain Stimulation, Dr. Charlotte Rosso identified an impact of functional connectivity on the resting motor threshold. Functional connectivity, briefly said, means a correlation in activation in different brain areas. We can quantify activation in brain areas roughly by measuring the bold signal. We can then extract the bold signal from, for example, the right motor cortex and correlate it with activity in the left motor cortex. If there's a high correlation, it means that both areas have a high functional connectivity. Dr. Rosso found that there was a positive correlation between functional connectivity between M1 and the dorsal premotor cortex and the resting motor threshold. She then suggested a model that explained variability in the resting motor threshold by age, coil-to-cortex distance, functional connectivity between M1 and the dorsal premotor cortex, and cortical gray matter volume. Going on from there, the aim of our study was to validate and replicate these findings on functional connectivity and the resting motor threshold. We also included the coil-to-cortex distance and age in our analysis, similar to the study by Rosso. We extended their model by including data from both hemispheres. Lastly, we also wanted to include the gray matter volume. However, I can't present these results today yet because they're still being analyzed. Our data set consisted from data from 2 studies with a total sample size of 38 subjects with a mean age of 37.5 years and roughly 50% females. Therefore, our data set is roughly double the size of the original publication. Our study protocol consisted of an MRI with an MP range and resting state fMRI sequence and an NTMS motor assessment. The NTMS motor assessment was conducted on a NextStim NBS5 stimulator. We'd identified the hotspot for the first dorsal iterosius muscle, so the muscle for the index finger on the hand on both hemispheres. On these hotspots, we defined the resting motor threshold using the Rossini-Rothwell method. Briefly said, this defines the RMT as lowest stimulation intensity with at least 5 out of 10 positive stimulations. The MRI scans were conducted on a 3 Tesla magnetron trio Siemens scanner with a 32-channel head coil. For analysis of the resting state fMRI functional connectivity, we followed the pre-processing steps used in the paper by Rosso and colleagues using the CON toolbox. We computed Roy-to-Roy functional connectivity using the human motor area template as an underlying atlas. This deviates from the protocol used in Dr. Rosso's study, as they used subject-specific ROIs drawn on individual FA maps. We chose our approach because we felt it reflects common usage of ROIs and common use of atlases in fMRI studies, and is a very standardized and objective way to identify ROIs. However, we picked our atlas so it matches the regions used in the original study very well and made sure that our registration of the atlas for individual subjects was matching the structure very well. The regions included in the atlas were primary somatosensory and primary motor cortex, supplementary motor area, supplementary motor area, pre-supplementary motor area, premotor area dorsal, and ventral. We also measured coil-to-cortex distance on the MPRH sequences, again following the protocol proposed by Rosso and colleagues. For statistic analysis, we followed two approaches. First of all, looking at a subset of the data for only the dominant hemisphere, according to the results proposed by Rosso, and then extending these results by looking at both hemispheres. We computed correlations between the resting motor threshold and the functional connectivity between atlas regions, age, and coil-to-cortex distance. We did the same analysis using linear mixed models for both hemispheres. Next, we tried to replicate the multiple regression model proposed by Rosso, including age, coil-to-cortex distance, and functional connectivity between M1 and the dorsal premotor cortex. However, it should be noted that in the paper by Rosso, this includes also the cortical volume of the dominant hemisphere. And finally, for both hemispheres, the same model as a linear mixed model. So let's take a look at the results. I'm not going to go into detail about the results from the correlations because there were no significant correlations except for the ones included in the regression model anyway. The regression model for the dominant hemisphere showed an effect of coil-to-cortex distance on the resting motor threshold. However, we could not find an effect of functional connectivity. This is supported by a Bayes factor suggesting that the null hypothesis is 5.8 times more likely than the alternative hypothesis. So this suggests that there is not only an effect of coil-to-cortex distance, but not functional connectivity on the resting motor threshold. Looking at data from both hemispheres, we saw essentially the same results. There was an effect of coil-to-cortex distance, but not of functional connectivity. And again, the Bayes factor suggests the null hypothesis over the alternative hypothesis by a factor of 8.3. So as you can see in these graphs, there is a nice positive linear relationship between the resting motor threshold and coil-to-cortex distance. However, no such relationship can be observed for the functional connectivity. So with those results, we then contacted the original authors again and asked them for their opinion on these results, as well as any ideas where our methods could have deviated from their protocol. And one thing that became apparent is that while it is not stated explicitly in the manuscript, Dr. Rosso conducted the MRI scans directly before the NTMS measurement. As I said, for our study, this was not crucial. So some of the MRI scans were conducted right before the TMS scans, some were conducted several days apart. Therefore, we figured timing of the MRI and TMS might explain our different results. And what we did next is we identified the subset of subjects that had the MRI directly before the TMS measurement and looked at their data. However, I should say that those were only 7 subjects, so all of these results should be interpreted with caution because they're highly underpowered. So now again, the results for the dominant hemisphere with only those 7 subjects. We still did not see any effect of functional connectivity on the resting motor threshold. And this is still supported by a Bayes factor favoring the null hypothesis over the alternative hypothesis by 2.38. So again, we see that functional connectivity did not impact the resting motor threshold. Similarly, for both hemispheres, functional connectivity did not impact the resting motor threshold, and a Bayes factor of 2.94 again favored the null hypothesis. So, taken together, we could not replicate findings of impact of functional connectivity, neither between the primary motor and premotor dorsal cortex on the resting motor threshold, no other functional connectivity values between other atlas regions. We could see the well-established effect of coil-to-cortex distance on the resting motor threshold, and we could see some effects of age on the resting motor threshold. However, they were mixed and not very strong. This might be due to some missing co-founders in our analysis, such as the cortical volume of the hemispheres. Lastly, we also did not see an effect of hemisphere on the resting motor threshold. So now we thought about why we could have different results than the study published by Dr. Rosso. And one possible explanation is that our analysis did not include the cortical volume yet. However, we think this is unlikely to have influenced our regression and mixed models, because also in the correlations there was no relationship between functional connectivity and the resting motor threshold. Next, our TMS system was different than the one that Rosso used. However, both systems were neuronavigated and the range of resting motor threshold variability was roughly the same. Then again, our MRI protocols and scanners were slightly different from the ones that Charlotte Rosso used. However, this is again unlikely to explain the different results, as any effect should be robust against slight changes in MRI protocols. Then, like I previously mentioned, there were some differences in timing between MRI and NTMS. However, looking at the results from our subset, it doesn't seem like this explains different effects either. And lastly, we can look at the different ROIs, which remains a factor of uncertainty regarding the comparability of our results. However, we would argue that our atlas matched the regions selected by Dr. Rosso very well, and we made sure that registration for the atlas onto the subjects was good. Therefore, again, it seems unlikely that this caused the different results, however, it cannot be ruled out completely. So, taken together, we could not see an effect of resting state fMRI functional connectivity on the resting motor threshold. We saw that coil-to-cortex distance remains the most important predictor of the resting motor threshold, and that the factor of age remains inconclusive, potentially due to missing confounding variables. And with that, I would like to thank my supervisor, Dr. Thomas Picht, as well as Fabia Roth and Leona Kawelke for contributing data to the study. I would like to thank Professor Carsten Finke and Darko Kumenic for helping with the resting state fMRI analysis, and Dr. Ulrike Krittner for answering any statistical questions I had. I would also like to thank the team of the BCAN for giving us access to the MRI scans and the MRI machines. And I would like to thank the Einstein Center for Neurosciences and the Medical Neuroscience Graduate School for funding my PhD and supporting me throughout. Lastly, I would thank the organizers for this great conference, and thank you all for listening.